Everybody loved it, but the FDA will look and said, well, you didn't meet your endpoint. Requirement that you set, you didn't meet that set, so basically it's a failure.
So, one of the other takeaways that I heard earlier was you want to control the variables. In the real world, there is so many different variables to these experiments. One of the things that we do now, which is a difficult process, is we try to assess the ROI of different projects. And the thing is, it's a theoretical. So we have a certain set of assumptions today of what the outcome is going to be for one project or the other. And we also have a theoretical budget of how much cost is going to be in there, whether it's production costs, software development, whatever it is that's related to it. The reality is when you start going into the real world, it's not a controlled environment. This is not a casino with a very specific set of rules and nothing else is changing. So while an experiment is running, our new idea, our new project, our new what product that we're trying to launch, there's 5, 6, 7 different factors that are influencing the behavior of that outcome at the same time. And I've historically used like factor analysis. Can you give us maybe not just the pre-experiment phase, but how do you think about influences of what you're trying to control for, and how we can think about maybe baselines? How do we analyze tertiary, secondary effects of the system, and how do you control for that? Because in the reality is, when you're doing human testing, you also have to go through this process as well, because you have to have double blind, these types of tests and stuff like that. So could you give us all some perspective on how those experiments are structured? What is a double blind test? What is p-value? What are the things that we should be looking for in how we structure these experiments, and how to think and put it in a way that we're not scientists, we're not PhD scientists here, put it in a way that we could potentially apply to our business, because you've been an entrepreneur for such a long period of time, how we could do this on a day-to-day basis as we're running different experiments in the company.
Yeah. So when with drugs and developing a treatment, I think it kind of often gets lost that the reason you're doing clinical trials and these experiments is really to please one big group, and that big group is the FDA. And the experiment that you design, you have to work backwards from. You have to get an agreement with the FDA as to what it is that is going to be the determinant for success. And you may think success, whatever success is in the real world in terms of treating patients, may not be the exact success marker that you agree with the FDA on. And that's because, yeah, you want to treat a particular cancer or a particular disease, and you want to see the patient get better, but that's a very sort of nebulous goal. It's not driven by data. And to quantify it, you have to come up with quantifiable measures. And this is what you want to get the FDA to agree on. And you'll notice, like, if you actually read the labels of drugs, they don't always match, even though they're treating the same patient population. And that's because the companies have agreed with the FDA as to what is the determinant for success. And you have to be really good at knowing the molecule that you're developing, or the particular way you're developing that, is going to address that particular indication. And so if you don't do that, then you'll design all of your experiments and you'll make mistakes. You'll think, oh, the results look great. This trial showed great results. Oh, and the patients were happy, everybody loved it. But the FDA will look and said, well, you didn't meet your endpoint. The requirement that you set, you didn't meet that set, so basically it's a failure. And if you want to use that data, you'll have to redo the whole study again, which basically means more money, same experiment, almost a complete waste. So what you're trying to do, and the reason these trials exist as phase 1, phase 2, phase 3, is because they're stepping stones. The idea is that at each step of the process, you're developing and learning about the data so that the final test you do will be a winner. Now, if you do your job right, you will design the experiments in a way that they will answer specific questions, so that when you design your final test, you can be as confident as possible with the FDA that it will meet that final endpoint. Now, if you get distracted, which is what sometimes happens, is you do an initial trial and you collect data and you try to collect the primary data, which is the primary endpoint, the thing that you're looking for to get success for, and you see other things, you may try to say, oh, this is more valuable now. Let me do the second experiment looking at this thing. And you'll start diverting multiple times. And eventually by the time you get to your final stage, you'll realize, oh, I'm far away from what I discussed with the FDA. And you won't have the backing of the data that you had in the early stages to justify it because you started diverting from that path. And so it's critical that you remember the end goal. You either modify the end goal based on real data, not based on small pieces of data. Because what typically happens, and this happened at Clearside, is you do a phase 2 type trial where you generate some efficacy and you say, wow, this efficacy looks great. And we saw this patient population, oh, they responded even better to that. Your collection of data was on 100 patients, but that patient population maybe was only 15 or 20. All of a sudden, investors start getting interested and they say, hey, if you could address that, that's a huge market. We would pay you a lot of money for that. Money starts to play a factor, and they say, well, design your next experiment based only on that 20 patient data. All of a sudden, you've lost the strength, the statistical data strength that you had, and you do the experiment and you say, oh, we're going to go after our drug being better than this other drug because we saw it work so well in these 20 patients. And you design it, and unfortunately, when you expand it and you do it in the 100, 200 patients, it shows no difference.
Let's pause there for a second because this very directly applies to how we do our experiments. One of the things that we used to do in the good old days, we would say, well, 100 people, and then 200 people, then 300 people. Um, what we've used now, like I said, is a Kelly criterion model of saying 2 million is our bankroll, but divided by the number of loans, say it's like, let's say it's $500 average loan. And so you're going to scale up that experiment. If it's starting to perform the way that you expect, then you keep scaling up, keep scaling up. Now, assuming perfection, meaning like every single point in the process works out, it takes about 3-4 months to get to the end point where you're like, we know this is successful in a large enough population. Now we can operationalize it for the whole entire company. But in that process, the starting points are very interesting because for us, we don't think we're going to lose all the money, right? It's not 0-1. But in many experiments, it's hard to figure out, okay, well, we're going to do it manually up front because, no offense to the software development team, you've got time, effort, energy, you got your own backlog, we've got all these other resources and projects that are going on. You then have to take away resources from one project to allocate them here, and the pipeline could take a very long time just to get the basic data. So when you're starting the experiment process, you have a limited amount of money, limited amount of resources. What's the minimum number of people, or how do you define the number of that minimal population to get started so you can say, hey, we're getting some traction, we need to move on to the next phase of the project?
Yeah, so a lot of that early stuff is based on kind of expectations. So the early trials are really geared towards safety. So what is the expectation on safety in terms of the side effect profile that you're trying to avoid? Like in the case of the current company, which is focused on peanut allergies, the thing that we're trying to avoid is an anaphylactic reaction. So there's a certain uh, population that you're going after. In this case, it's peanut allergic patients. And a lot of that is based on historical information of the likelihood of somebody having an allergic reaction and determining a population size in which you can detect the incidence at, say, a certain confidence level. So if you want to be able to detect that with 80% confidence that you're going to detect at least one occurrence in your population, then we had to figure out that you need at least 20 patients to have 80% confidence. And if we have that 80% confidence in this first trial, then we can design the second trial based on that data set. So our first trial is with 40 patients and also the number of doses. So it's not just a matter of a single dose of exposure, but multiple doses of exposure. So you can say, I can have this level of confidence with this dose because I gave everybody this dose, but maybe the highest dose, I only gave half the patients. So then you say, well, I have less confidence that we detected all the safety events at the highest dose. And so those are done by biostatisticians. They take a look at the representative population, incidence, and historical information in other trials and determine that, hey, you need a certain number of patients to detect with this level of confidence. And then you have to determine with the resources you have how much confidence you want to have at this point in the game.